Assalamualaikum warahmatullahi wabarakatuh. We're from the fourth group. On this occasion, we're gonna present our discussion about production of plant antibodies as plant protection products. First, let me introduce the fourth group members. Nugroho with student number 093, Trisna Prasatria 094, Ikhsan Lazuardi Rachmansyah 100, Dilla Islami Salsabila 102, Mohammad Daffa Herda Pramudya 104, Muhammad Yusuf Effendy 105, and Karenzky Yehezkiel Putra 108.
Plant antibody. A plant antibody is an antibody that is produced by plant that has been genetically engineered with animal DNA and encoding a specific human antibody known to neutralize a particular pathogen or toxin. Various plant defense structure include the amount of quality of light and particularly the epidermal wall structure cell, the size, location, and set of stomata and lenticels, which on the surface of fluid and leaf can prevent the information of a layer of water that can be a place for pathogenic fungi to germinate and bacteria to multiply. Likewise, this layer of or on the surface can also repel water, greatly reducing the chance of infection. The thick and the hard epidermal cell wall is difficult for pathogen to penetrate. Similarly, the thickness and hardness of the tissue cell wall will make it difficult for pathogens to grow. There are plant defense structures that are formed after infection. Examples are the formation of coat layer, adhesion layer, pilosis, and gum formation. Similarly, there is also an attempt to work height that penetrate the cell wall with materials derived from the webbing of the cell wall. Against obligate pathogens, can often react hypersensitively, namely the rapid death of cells around the infected are so the pathogen is confined to the tissue and thus does not to get food. The sooner the host cell dies after infection, the more resistant the plant will be to infection. Biochemical defense for resistant to pathogenic infection, chemical defense mechanisms play a more important role than structural ones. This is evident, among others, by the absence of pathogens in plant cells that do not have an inhibitory structure.
Antibody. Some chemicals that inhibit pathogens are released by plants before an attack occurs. Some are released after introduction by the invading pathogen. Examples of the first include toxics, substance found on the surface of red skin onions, organic acid, a phenolic compound that is toxic to many microorganisms, is more abundant in resistant potatoes than those susceptible to scurvy, caused by Streptomyces scabies. Similarly, the roots of potato varieties resistant to verticulum contain more organic acid than the roots of susceptible varieties. Examples of the latter include production of toxic substance around damaged tissues or formation of protective layers such as callus and cork. Most of these inhibitory compounds are phenolic compounds, as well as phytoalexins. These phytoalexins are not present in healthy plants, but are produced after stimulation by pathogens, medical or chemical injury, alteration in protein synthesis in plants that can cause local resistance or a thick layer around the site of infection, the emergence of compounds that are not easily broken down by pathogenic enzymes that will attack them, phenolic compounds that inhibit pathogen enzymes.
antibody structure. There are two antibody structures. The first is FIB structure. The FIB section that has variability to recognize antigen is found in the variable region or region V. This region is important in antigen recognition so that it becomes the focus of research in developing monoclonal antibodies for therapeutic purpose as well as designing antigen used as vaccine. Region V consists of 6 parts of the complementary determining region or CDR, namely 3 from the HC subunit and there from the LC subunit. Amino acid residues that are in direct contact with antigens are called paratopes. The surface of the antigen is in direct contact with the antibody is known as the epitope. The second is FC structure. The lower part of the immunoglobulin is called the FC region or region. This FC consists of a pair of HC protein subunit with a constant or fixed structure. The FC part serves as an effector function and play a role in determining the half-life of the antibody. The effector function of FC includes being involved in cell-killing mechanism through the interaction of proteins and carbohydrates with FCY receptor and the complement system. The function of determining the half-life is related to the interaction of FC with FcRA.
Antibody production. Antibodies that come from a single immune cell are called monoclonal antibodies or MABs. MABs have become ubiquitous tools in biomedical science and medicine. They have been used to research, diagnose, and combat diseases. Monoclonal antibody production can be carried out in vitro or in vivo by growing hybridoma clones resulting from the fusion of lymphocytes with myeloma cells, so that specific monoclonal antibodies for detecting LTV can be produced continuously. The lymphocyte cell used were derived from the spleen organ of biopsy mice that had been injected with antigen from LTV. The immune response shown by the formation of antibodies by lymphocyte cells can be generated by injecting experimental animals with antigens known as potential antigens that uses monoclonal antibodies that can detect LTV will help control spread of tumor disease, especially in areas of rice production center in Indonesia.
In 1925, Kohler and Milston introduced the technique of culturing antibody production cells through fusion with myeloma cells, so that they can produce homogeneous antibodies continuously, have specific serological reaction, and have certain biochemical characteristics. Hybridization of antibody producing lymphocytes with myeloma cells or malignant myeloma cells produce hybridoma cells that combine parental traits and the ability to produce or secrete specific antibodies and continues to proliferate. Cloning and further selection of hybridoma cells allow the production of MEBs with a same specificity or identical and effectiveness as desired against certain epitopes on antigens used to immunization in Jordan 1990. Recently, MAB production techniques have been adopted for the production of plant pathologic MABs. MAB has been used to detect and identify plant pathogens, including virus, phytoplasmas, and bacteria, from Converse and Martin in 1990, and McLaughlin and Chen 1990.
Antibody production. The advantage of MAB production technique include A, being able to produce a relatively unlimited number of antibodies. B, the ability to preserve specific AB production through cryogenic storage of hybridoma for an indefinite period of time. C, the ability to produce and select MABs for almost all antigenic determinants used for immunization are impure or mixed. Jordan 1990, 1990B, DeBrew 1990. Hybridoma technology for MAB production has been applied to plant virology and bacteriology. MCAB is very useful for the identification of qualitative assays on bacteria and plant viruses. For seeing the degree of similarity between members of various groups of viruses and bacteria, and for studying the structure and function of gene production. Preparation and characterization of MABs for pathogens and pathogens can be carried out. Various publications of MAB production techniques can be used as reference material. MCAB has been produced for a variety of plant pathogens, including viruses, phytoplasmas, and bacteria. Thank you for your attention. That's our presentation. Thank you for watching. For the question can be sent through the WhatsApp group. Thank you for watching. Wassalamualaikum warahmatullahi wabarakatuh.